Welcome, everybody. We'll just give it a few seconds as you all trickle into the meeting before we start. Okay, thank you everybody for coming. This is our first Distinguished Speaker Series of the spring term. We do have two more scheduled for April 13th and April 27th, so we hope you can attend those. Today's speaker is Dr. William Coon, who will present the science of sleep, learning, and memory, how hacking your sleep can help you learn better and remember longer. Dr. Coon is a sleep scientist and neural signals engineer at the Johns Hopkins University Applied Physics Laboratory. He earned his PhD in brain-computer interfacing and neural signals analysis at the National Center for Adaptive Neurotechnology in Albany, New York. He is trained in the science of sleep, circadian rhythms, and learning, starting as an undergraduate at McGill and Brown universities and culminating in 3 years of postdoctoral experience at Harvard Medical School. In Dr. Coon's work at the Applied Physics Laboratory, he fuses sleep science and brain-computer interfacing engineering, developing real-time closed-loop systems that measure and interact with the brain's nocturnal information processing functions. So, without further ado, I'd like to welcome Dr. William Coon. Thank you so much for joining us this afternoon.
Thank you, Zoe. Thank you, everyone. I can't tell you how excited I am to be here and have an excuse to wax excited about all of these seminal sleep studies that have gotten me so interested in this topic that I made a career out of it. And I hope I could share at least a few practical takeaways that you can use in your own lives too. Generally in the sleep community, people like to point out that this is not a new idea, the relationship between learning, memory, and sleep. And one of the more often touted quotes is from the rhetorician Quintilian here, who noted that a night of sleep strengthens the memory. And the concept is pretty simple. If you learn something and let some time elapse and are tested on it later, hopefully from the process of learning and studying, you have some performance improvement. But if the intervening time is actually sleep instead of just passive passing of time, you actually perform much, much better. And we're going to dive into the details of that in the next few studies here. Just to orient where we're going, I wanted to start with a very, very short primer on what sleep looks like and how we measure it, so that we're all oriented to the different types of sleep stages that we're going to be looking at, each of which really has their own specialized functions, and together they interact to do more than the sum of their parts in terms of information processing during the night. We're going to spend the bulk of the talk today on some of the seminal papers and studies that relate sleep learning and memory to sleep stages and their particular functions, with some practical takeaways along the way. And then that's going to lead up to the last section where we're going to talk about all of the fun stuff, how we're interacting with sleep, with BCI technology and other similar systems to engineer sleep and even maybe upgrade your brain. So this is what a good night of sleep might look like. We'll start over here. In a typical night, you go through multiple stages of sleep that cycle repeatedly every approximately 90 minutes or so, starting in wake, dropping down into deep, what we call slow wave sleep, and up to lighter stages. Slow wave sleep diminishes as the night goes on, cycle to cycle, and becomes progressively replaced by more and more stage 2 sleep, which is kind of like a engine running in neutral with some added benefits. And also these periods of REM sleep, rapid eye movement sleep, which many of you have no doubt heard of already and is typically associated with things like dreaming. In order to get one of these maps of sleep, the traditional measurement of system is called polysomnography, which is a composite of 3 different electrophysiological signals: electromyography for muscle tone, electrooculography to capture those rapid eye movements, and EEG is where most of the interesting stuff happens, brain signals. And there are grapho elements in the EEG that really pop to the eye, salient features that allow one to classify periods of sleep according to the behavior of those elements over time. So for example, if you look at somebody who is first getting into bed and closes their eyes, your sensors over visual cortex in the back of your head will show this prominent, strong alpha rhythm, about 10 cycles per second, 10 hertz. And the disappearance of that alpha is your first indication that you have tipped over into stage 1 sleep. So that's when you're falling asleep from being awake. Slow wave sleep is probably the easiest to identify because it's characterized by these just massive, low frequency, high amplitude, slow waves in the EEG. And what that reflects is hypersynchronous, really hyper coordinated activity that travels the surface of the brain, the cortex, in waves. And we'll delve into some of the functions of those in a little bit. Before you get into slow wave sleep, you typically transit through stage 2, and this is where you do spend a lot of the middle and later part of the night. Stage 2 is an interesting mix, and it's marked mostly by these kind of transients that appear. For example, an isolated K complex, which in some ways can be thought of as an isolated slow wave with some debate about different functional implications. And the classic sleep spindle, this enveloped oscillation that looks like a spindle of thread. REM sleep is, of course, marked by the eponymous rapid eye movements, which you can see here. The EOG sensors are just to the left and right of each eye and a little offset vertically, and that captures the voltage potential difference reflected from the front of the eye to the back. There's actually a highly negative polarity in the back in reference to the front of the eye. So the movement of the eye in the orbit will cause your electrical sensors, same ones you use for EKG, EOG, EEG, to pick up these big deflections reflecting eye movements. So we can get those electrophysiologically. Now, again, to generate that map of your sleep over the course of a night, typically and historically, since the 1950s, you would have a trained expert, a physician, a scientist, or a technologist who has learned how to interpret these signals and then apply explicit objective rules, but there's some subjectivity in the interpretation. So there really isn't a perfect ground truth label. Nevertheless, this is a consistent way of categorizing sleep stages, and people can be trained up to be about 85–90% in agreement with each other about what stage you're in. Obviously, this takes a tremendous amount of time, so we could say that this is how sleep was measured then, and these days it should be no surprise that we're using automated decoding algorithms at this point. It seems like everybody and their cousin has some automated decoder that can classify sleep stages, usually offline, although we're making some really fun strides translating that to online versions. We'll get to that later. This is typically combinations of early convolutional neural network layers to pick out those grapho elements that pop to the eye. CNN's really good at that kind of stuff. And then the temporal dependencies across longer scales of time are captured by recurrent neural networks. This is similar to how we did it with colleagues at the School of Medicine at Hopkins here. In some of our recent work, though, just like everybody else, we're moving from RNNs to transformers and multi-head attention implementations. And if we have time to get into it at the end, there are some interesting trade-offs. Transformers don't obsolesce the CNN RNNs, especially in wearable systems that are supposed to be lightweight and resource not intensive. Nevertheless, at this point, we have basically obsolesced the trained expert scorer, which really facilitates big data studies and rapid decoding. All right, primer done. Let's dive into some of the seminal studies here. I'm going to refer back to a few conceits of the lecture. This is just one possible taxonomy of human memory, with major divisions in declarative, what I can capture and express in words, and procedural or implicit, things like learning how to ride a bike or sail a boat or drive a car with a stick shift transmission that doesn't necessarily translate to words well, but can be objectively measured in a performance sense. The declarative memory system is traditionally associated with the hippocampus, said to be hippocampus dependent. And that's where we'll start today. A lot of the stuff that happens, in fact, almost all of the stuff that happens in the brain during sleep is different ways of communicating between multiple brain systems. In this case, the hippocampus, which you see buried in this deep structure. You have one on the left and one on the right side of your brain. And the cortex, the outer rind, just the surface of the brain here, where all your sensory perception happens during the wake, rumination, cogitation, default mode network activity, and presumably most long-term storage is cortical destination. The hippocampus is responsible for rapid acquisition of new learning, and we'll see that during the night, it basically plays the role of teacher to the cortex to stabilize and consolidate new learning into long-term memory. That's what we're going to look at in this first study here. In this study, they had participants come into a lab and learn to associate some pictures, essentially. And they let some time pass, up to 3 months. And they were tested at intervals in between by presenting pictures and asking, have you seen this before or not? Or in the other version of the task, is this paired with this or not? Looking at the neuroimaging in this study, they see this interesting pattern where over time, as people learn the task and are tested, they learn, or their brains rather, recruit the hippocampus less and less as the memory becomes stabilized and hippocampus independent and relies only on cortical activation for recall. Really quite nice study there. And digging into the architecture, they find that it's slow wave sleep, which correlates with the degree of improvement, sleep enhanced memory, the benefit thereof. This is the exact memory circuit that I spent my postdoc years working on when I was up in Boston. And it's basically a coordinated dance of these nested oscillations that allow the two major players in this operation, cortex and hippocampus, to talk to each other efficiently. This is brokered by a middleman, the thalamus, that provides an intermediary signal, the spindle actually, to kind of package and isolate different reactivation events represented in the hippocampus and propagated out into the cortex in these about 1 hertz slow oscillation windows, reading frames almost. And these multiple temporal scales are helpful for capturing spatial and temporal dependencies at different scales. So slow wave sleep improves performance. There's our second conceit, referring back to our hypnogram. And this is a 2-stage process. We talk about being systems level. And this implies that there is some memory reactivation. Obviously, some type of processing is happening during sleep. It's active. It's not just passive passage of time. And that's what we're going to delve into here, pivoting to animal models. The following work was shared with my primary colleague in Boston, Matt Wilson, who is now at the Peichauer Institute for Learning and Memory at MIT. We'll talk about my other collaborator, Bob Stickbold, later in the talk. Matt has done some absolutely brilliant work on information representation in the rodent brain. We mentioned that the hippocampus is an older structure. It's highly conserved, especially within mammals. So, in fact, a large proportion of the rodent brain is hippocampal circuitry that is in many ways identical to the way our hippocampal circuitry is laid out as well. It makes rodents in a maze a really good model to do this type of research. A lot of you would probably already know about hippocampal place cells, which are these special types of cells in the hippocampus that fire or respond specifically to certain locations in space. And what you can do with that, if you look at a population, you zoom out and look at a few of these together, you can actually map out trajectories through space and even follow along as the animal traverses some exploration environment or a linear track maze, and map out the firing patterns to decode the trajectory. If you're looking at populations of cells, the way you do this is you record from, again, a large population. You can then kind of organize through a process that we won't get into the details of spike sorting, but you can basically label a cell with its corresponding location in space. And here that's shown as a color. So one cell fires as one dot on this map, and one color is the location it encodes, or one color is rather the cell. You can then order these firing events and look at the sequence of their activity, and that tells you about where the rodent is moving in space. So this is the progression across a maze. That gives us a template that we can then search for in the sleeping animal. And there are correlative mathematics we can apply to sift through a lot of noise and identify periods where these trajectories are recapitulated during sleep. And sure enough, these occur in the context of our friend, the hippocampal ripple. So this oscillation co-occurs with the replay trajectory we see in rodents. So the actual reactivation of the exact same patterns of activity learned during the day, nested in this ripple, packaged, bundled, and organized by your spindle, and propagated out to cortex. I'm not sure if the sound comes through on this, but the visual is really all we need here. This is actually a video from Matt's lab looking at one animal. There we go. And you'll see in real time the activity popping off here. And what you're looking at, this arrow that pops up. Oh, and did you catch that? It ran backwards. This is nothing fancy. This is maybe 10 years, 15 years ago, looking at a very, very simple linear decoding model that identifies where the rat is in space based on the activity of these cells here. And occasionally you'll see that pattern also shoots around in reverse, often when the animal stops and pauses. This turns out to be really important. The animal, when they're pausing, they're actually thinking about where they want to go and what they've learned over time. The backward sequence is something that we observe during sleep also. And I like to think of it, it makes sense to me when I think about this animal is learning goal-directed behavior that's mediated by some reward acquisition, essentially. And if you think about most reinforcement learning models that operate on Bellman equation style updates that propagate credit back in time, this is not back propagation, of course, this is credit in the reinforcement learning sense, from some end goal back through locations. So it does make sense in a way to reactivate patterns starting with the goal or objective and then map them back to what worked to get you to that objective. Now, here's where things get really crazy cool. We can, of course, do the exact same decoding when the animal is sleeping. And you probably just saw the backwards replay just fire past there. This is no longer just the domain of animal research. This kind of blew my mind when it came out a while ago. But nowadays, we're actually starting to see the same type of thing happen in humans. This science paper came out in 2019 and made a really big splash in the community. You have rare opportunities sometimes to work with patients, usually who are having brain surgery and need brain mapping prior to that surgery, either to remove a tumor or dead tissue or to treat epilepsy. And while they're doing the brain mapping, you can do some research studies like this. So this particular subject had electrodes in their hippocampus and in their cortex. And the clever experimental design component here was there was no stimulus response in this design. This was a free recall. They were asked to passively view a bunch of pictures while recording the neural response to those. From that, they could tell which cells were selective to visual imagery, essentially. And then they could ask the patient to sit and ruminate and free recall, just like that rodent who is pausing to think about possible futures in the maze. In this case, the individual pauses to think about what they've just seen. And they see a beautiful canonical increase in the probability of these ripple events that we know are related to transferring information from hippocampus to cortex in a replay event. So it gets even cooler when you start applying things like classifiers to the neural activity. During free recall and during sleep, you can identify periods of increased rippling activity and then decode the likelihood that they're looking at one image versus another, which is getting scarily close to being able to decode dreams. I say that, and then this work comes along. This is brand new work, just came out in today's world of GPT-4 and stable diffusion and similar models like that. You can start to do some really mind-blowing science with these things. So this is a similar concept, taking a diffusion model, which is, I like to think of it as learning an entropic minimum and backing that entropic process up to a more structured space. So it kind of learns how like ideal gas laws in a closed volume spread out to minimize entropy. It learns to map that process back to an ordered state. The analogy translates to visual imagery in this case. So you can start from disordered noise, brain signals, and then map them back to a structured representation, in this case, of images. And these guys were successfully able to train their decoder model on awake passive viewing and then really actually reconstruct the experienced replay events in the participants' dreams. So this is just the cusp, and I expect that this area is going to just blow up with a lot of exciting opportunities in research. Yeah, it's crazy close to mind reading. We're really excited about this. So learning is replayed during sleep, and this is important for locking in long-term memory. And so far, we've hit on a few different types or different stages of sleep that participate in this process. Slow wave sleep, really good for declarative memory. Think about studying for a test, learning new pairs of things. I always think of organic chemistry the way I used to study for that. Here we go, our REM sleep and our slow wave sleep. Some practical takeaways. This is not news, but for me, seeing the actual science convinced me that a lot was going on, really translated to changes in how I behave in my life and how I even do things like prepare this talk. Study, rehearse during the day, and then let yourself lock that in overnight. If you compare 3 hours of studying in a cram sense back to back, likely you can do the same amount of learning spread out over 3 days in 15-minute chunks. So maybe 45 minutes or an hour instead of 3, and let your sleep take care of the locking in of long-term memory. As long as you are diligent about spreading little pieces out over time, you do yourself a favor and actually create a whole bunch of free time that you wouldn't have had otherwise if you were sitting there trying to force it into long-term memory purely through rehearsal cram sessions. So this is like studying for tests. I'm going to switch gears a little bit into procedural learning. This is implicit learning, things like motor skills, playing an instrument. And that's exactly why this study is near and dear to my heart as a trumpet player. I've actually changed how I practice the trumpet because of this research. The study actually originated with Matt Walker here, who is, yes, the author of the now well-known Why We Sleep book. This is back when he was a postdoc with Bob Stickgold at Harvard Medical School, Beth Israel in Boston. Matt is now running his own shop along with Bob Knight and others at UC Berkeley on the West Coast. But this idea of rehearsal, in his case on the piano, occurred to him, and they developed a task to examine this. This Guitar Hero-ish thing we're looking at on the left is just a visual depiction of learning a kind of an awkward sequence to touch. You map each of your 4 index or non-thumb fingers to these numerals, and you learn this sequence 4, 1, 3, 2, 4. And your job is to learn that and in 30-second bouts, repeat it as fast and as accurately as you can. So speed goes up over time, accuracy goes up over time, and you can see that in these nice learning curves, which give us a nice classic saturation point with an asymptote. And at this point, you're not going to be getting any better at the session. So we cut it off after about 12 training sessions. And the 2 colors are simply to show that learning is basically the same if you do this at 10 in the morning or 10 at night. We see differences when you look at the 12 hours that follow the learning. Perhaps unsurprisingly by now, you see no significant improvement if you just let a day elapse in between learning and testing. But just this massive improvement. This is just such it's a widely replicated, big effect size, probably the best replicated finding in the sleep and learning literature. But at night, your brain is really, really busy automatizing all of this sequence learning that you've done through the day, all of that takes a lot of driven, deliberate effort cortically becomes replayed, consolidated, and presumably integrated with circuits like the basal ganglia, which are typically associated with this type of automaticity in motor skills. Also not surprising at this point, it does correlate with the amount of sleep that you get. This time, though, we're talking about stage 2. So declarative memory is your slow wave sleep. Slow wave sleep, by the way, is the one that is homeostatically driven. It's the one that you need most of if you've been sleep deprived, and the one that, well, we'll come back to that. Stage 2, we mentioned, was filling up a larger percentage of your time as you proceed across the night period, interleaved with periods of REM. So your brain is shifting gears, working on different memory systems and different types of learning in different sleep stages before passing the baton again and cycling through the same process. We mentioned declarative memory being hippocampus dependent and procedural nominally being something quite different. This work done by a colleague, Anna Shapiro, who is a contemporary with me back in Boston and now runs her own shop at Penn, doing some of the most mind-blowing modeling work on artificial intelligence that sleeps to replicate some of these processes anywhere in the world. Her lab is growing rapidly. If any of you are interested, I'm sure she would be pleased to hire more grad students. But in this study, she looked at the exact same procedural learning task, but in patients that had a hippocampus and patients that for some reason lacked a hippocampus. Usually this is when egregious epilepsy can only be treated by removing large portions of the brain. Sometimes you end up having to remove some hippocampus. And in this case, what she found was that procedural learning was dependent on this hippocampal reactivation during sleep. So here's your amnesia MTL, medial temporal lobe. This is the people without the hippocampus. They experience no bump in their learning as a function of sleep, whereas normal human beings with their fully intact hippocampi do just fine, show the normal sleep bump. This is really interesting because it tells us that although the hippocampus has a more restricted role during wakeful learning, it actually becomes the major power broker in this nocturnal sleep enhanced learning process that we've explored here, and actually subsumes some responsibilities of other circuit systems and memory types and modalities normally outside of its wheelhouse. The story is always more complex than it first appears. So in this case, procedural memory, scales on a piano or a trumpet are associated with your stage 2 non-REM sleep. And at this point, I think the implications are fairly natural. I actually do use this when I practice. I practice in the evenings and I space it out. And sure enough, if I test myself and in the morning after breakfast, something that I've just hammered into just noise the night before, suddenly it's like my fingers are playing the scales for me. If any of you are musicians, I urge you to try this out, because it is one of the few scientifically reported effects that is large enough that you can actually experience it yourself subjectively. Really, really cool. We mentioned the story always gets more complex. So this part is going to be a little bit interactive. This is a verbal memory task in the vein of a declarative memory learning task. And I'm actually going to ask you to play along with an example of what the subjects in this task were instructed to do. You're going to see some words appear on the screen one by one. Pay attention to them. Your job is to learn them. And yes, I will test the group at the end of the presentation here. All right, get ready. Go. Okay. The way this webinar is set up, I can't see the audience. So typically I would ask for hands raised, but I'll just trust that you're playing along at home here. I'll start off by asking if this was a word you've all seen. Probably the answer is no. Table. Probably the answer is no. Doctor. Many people will say yes. Many people would be wrong in this case. It makes sense in retrospect, since it's obviously related to the general gist of all the words that you did see: nurse, medicine, health, hospital, physician, etc. But you actually never saw the word doctor. This is a really interesting and subtle finding here. This type of paradigm is called a Diess-Rediger-McDermott, or a DRM task. And if you look at performance on this task, in this sense, we're not looking at improvement, we're actually looking at less forgetting. So they're given enough of these that they can't possibly remember every single one. That's intentional, it's the study design. And what you're measuring is how much less they will forget based on your experimental condition, in this case, wake versus sleep. Their recall for the actual words that they saw, they forget less with sleep. But those gist words, the ones that they actually didn't see, but thought that they remembered, huge boost. Not only did they retain them all, they actually performed better. They switched from less forgetting to actually improving their performance in these gist words. Make things even a little more complex. We've been looking at all of these nice positive correlations so far. If you get more of this kind of sleep, it does better declarative memory consolidation, procedural memory consolidation. In this case, we're looking at a graph that tells us there is a negative correlation. More slow wave sleep you get, the less of those veridical words you're able to recall. So on a first pass, it looks like this is not a good thing. You want to remember those words. You were told to study and remember them. Presumably they have some value for you in the future. But viewed another way, it's probably not the most efficient thing for a brain to do to remember every little detail. In fact, it's a really efficient compression mechanism to compress or extract or distill some kind of gist that contains all of the pre-learned associations that could pattern out into each of those individual words, but can be easily retained as just one representation, one word: doctor, physician, hospital. So in that sense, in a world where we cannot possibly remember all of the stimuli we encounter, and we have to learn to select what we want to retain and rehearse that and pay attention to it, and I got interrupted here. Okay. Yeah. Yes, so efficient compression. Yeah, so it actually can be seen as quite an advantage to be able to switch from remembering all of these irrelevant details into just the key facts that you need to retain. We won't spend time on this slide, but just to say that a lot of the stuff that we've gone over so far is true for naps as well as overnights. And I've actually found it really helpful in my own work, especially if I'm reading a really dense article that I want to retain, to sit down, go through it in detail, and then take a nap for half an hour. Half an hour is a good target because you will get some non-REM sleep typically in that case. If not, quiet wakefulness actually does allow you to rehearse, and for some of those reactivations, the rumination that we saw in the rodents and the free recall experiments, that to occur. So even if you don't feel like you got to sleep, you're going to get some benefit out of it. And you cut off your sleep before you get into deep, slow wave sleep, which would make you feel groggy. And that's when naps become disorienting and counterproductive. But a half an hour quick nap to lock in what you've just read or learned, that's something I deploy all the time. So in this case, we suspect the brain is doing some very efficient gist extraction to compress information learned during the day. And this does highlight that we can come up with all of the taxonomies of memory types we want, but things aren't as cut and dry as that, or as clearly defined. There's going to be sloppy boundaries between the two. Okay, I think we're on basically the last study before we get to upgrading your brain. So this plays into an exciting theme. Okay, I'm going to do two studies here, actually. In this task, you are instructed to learn to predict the weather, sun or rain, based on a card that you're presented on a screen. And in this case, you're given the weather report, the prediction card on the left, just an arbitrary pattern of shapes, and then the actual weather. Okay, so when I see this diamond pattern, that means it's going to be sunny. And maybe when I see these patterns, it means it's going to rain. Okay, so I'm starting to pair these things up in my mind. Then we get our diamond pattern again, but this time it results in rain. This is confusing, but that's by design. The rules for this task aren't deterministic. They are, as you've guessed from the title, probabilistic. And the association with one weather pattern or the other is dynamic over the course of the task, meaning the probability of one or the other is assigned in these decrements here. Now, of course, this isn't something that somebody could report to you, even though behaviorally they're able to learn this task quite well. If you ask them, what is the probability of rain given the pattern of circles? No one's going to be able to say, well, it's 41 or 42% or 40%. But empirically, you can test them and they will demonstrate this learning and that they have absorbed these rates or these probabilities well. And no surprise, again, I'm going to tell you that sleep plays a big role in this type of inference learning, type of extracting complex patterns or distilling from complex patterns a simple compressed representation, often involving some kind of insight. And in this case, it correlates with REM sleep. Insight and REM sleep is one thing that I really hope that you'll take away from today's talk, because it's one of the most exciting things that your brain can do for you, and you get it for free just by having a brain. So this hints implicit rule learning and maybe some insight, but I'd like to share with you the study that really made me decide I wanted to spend a career investigating how the brain, sleeping brain, processes information. This is a paper, a Nature paper from 2004 that blew my mind at the time. Simple task. You're given a panel of numbers here. You'll notice that there are 3 numerals, 1s, 4s, 9s, and you have a simple job. Given a sequence of these, you're going to be given a whole set of these in the experiment. You apply some rules. Take the first 2 numbers. If they're the same, carry the number forward. Now compare that carried forward result to the next number of 4. If they're different, you carry forward the 3rd number, the one that wasn't in the pair you considered. Now 9 and 4 is a pair, which means we should carry forward a 1. Now 1 and 9, so we should carry forward a 4, and so on and so on. And your objective is really just to get this last digit. You're given a whole bunch of these on a piece of paper, and you need to turn in the answer at the end. You don't need to show your work in between. You just need to get to that last digit. Many of you, being as keen as you are, have already noticed there's some symmetry to be observed. And you've probably also discerned that you can shorten the process even more. Instead of paying attention to these 3 and flipping them, you can just keep this second one and carry it towards the end. The experiment is set up so that all of the sequences will do this, which means that if you discover this trick, you no longer have to do all of the individual sequence operations. You can just do the first and the second and retain it. And the beautiful thing about this is that you can actually measure the time or identify the time when this insight happens, because the amount of time it takes to complete the task will suddenly drop. And that discontinuity tells you empirically where somebody has suddenly had an aha moment. This turns out to be, I'm noticing the time, so I'm going to speed up a little bit here, highly dependent on REM sleep. Subjects who had more REM sleep were more likely to have this type of insight. I'm gonna have to go through this again. And this gels with all kinds of anecdotes from science. Auguste Kekulé, in the description of the structure of the benzene ring, he claims he saw in a dream. Mendeleev in the periodic table of the elements, also attributed to something he saw in a dream. And again, this is a finding that I have applied in my life. If I'm stuck on a problem, if I have writer's block, or when I'm preparing talks and I'm not sure how to structure the narrative, I'll spread that out over several days. I'll go to sleep and I'll know that in the morning, I'll have all of these ideas about how to reorganize things or move slides around to tell a more coherent story or what have you. This has worked time and time again for me. And it's another one like the scale learning and stage 2 sleep that you can actually experience yourself if you're deliberate about it. So this is another one I really encourage you to try out because it is very, very cool. Due to the time, I'm going to skip going through this study, but where we were going with this was simply that naps work as well as overnights in some cases, in most cases actually. And in this study, they looked at the content of dreaming. We talked about insight being related to REM sleep. Well, dreams don't always happen during REM sleep. They also happen in non-REM sleep. And in this study, they show that those subjects who dreamt about or reported dreaming about the maze that they had been tasked with learning, a spatial navigation task, if they had images of that in their nap period, they did much better. So there may actually be a functional role to your dreams. Okay, let's get to the fun stuff. Hacking your brain. Okay. There are a lot of ways to interact with all of the processes we've just gone over together. And the oldest and most effective right now is slow wave stimulation. We know that slow wave sleep correlates with memory improvement. Spindles correlate with memory and procedural learning. Different other graphical elements like K complexes, isolated slow waves participate in this learning process. And we saw a whole bunch of positive correlations where more slow waves was better. And it turns out that the slow wave system is highly susceptible to being encouraged to produce more slow waves. But you can't just blast off stimuli at some 1 hertz frequency independent of what's going on in the actual brain. You have to, one, apply your stimulus only in the right sleep stage, stage 2 or stage 3 non-REM sleep, and you have to time it so that it coincides with ongoing rhythms. It's kind of like you're measuring the meter of the brain's rhythm and timing your stimuli to follow where a slow wave would appear. And with that process, you can either through a transcranial electrical stimulation, it turns out that actually just clicks auditory stimulation works better than electrical stimulation. A lot of the electrical stimulation stuff, by the way, is a little snake oily, although there is something there. Auditory stimulation works best. You can turn one transient slow wave into 2 or 3. So you can significantly increase the number of slow waves you have. And there have been a number of studies that show that this type of procedure can actually enhance your learning, upgrading your memory. So you can enhance the activity of your already innate ability to process information while you sleep. This does require real-time processing. This is where the BCI component comes in. We're doing this type of work at APL right now and developing our own wearable system with our own decoders that operate in real time to decode accurately in real time the user's sleep state and then perform that millisecond precision timing for stimulation to increase slow waves. This is a big tent. Everybody has been looking at slow wave stimulation for the past 15 years or so for all kinds of purposes, increasing getting you to fall asleep faster for insomnia, targeting declarative memory, targeting motor memory, generalized synaptic plasticity. This is a really hot topic, really hot field, and is sitting there waiting for other young engineers to get as excited about it as we are and start manipulating brains. There are other ways to increase slow wave sleep. The slow wave stimulation route is one way to increase the number of slow wave events, but it doesn't always translate to increased wall clock minutes spent in slow wave sleep. If you want to extend the amount of time you're in that sleep state, there are ways to do that. And we have an ongoing project in collaboration with the School of Medicine here, using our own device wearable system to do that. I'm currently under a little bit of an NDA situation, so I won't tell you what the secret sauce is there, but this has been just a delight of a project to put together. We can actually run an entire stripped down Linux kernel on a processor the size of a stick of gum. This is the latest Arduino. It's running a Nordic Semiconductor IMX8. It's a quad-core processor, ARM A53, with an M0 to boot. Anyway, in any case, it's actually running a customized, completely lightweight, sleek, no extra fat on it, Linux kernel that we compile with our custom sleep mod software for this project. And we hope to be collecting data in subjects in the next few weeks, actually. Now, I'll leave you all with another really cool area that is much more recent, although the phenomenon was described almost exactly 10 years ago. The research has only recently been picking up into higher gear. This is called targeted memory reactivation. This is work by Ken Paller. and his group. I think they're in Chicago, and Ken is a memory guy. He actually hadn't been involved in sleep research until he got turned on to this idea he had about reactivation during sleep. And he just burst onto the sleep scene with his findings that you could actually encourage the brain to reactivate certain memories and not other memories with carefully timed interventions in the overnight period. So in this case, a study participant is given a reading assignment or learning objective, and different elements of that learning objective are paired with scents or sounds or other completely unrelated sensory stimuli that form an arbitrary link, an arbitrary synaptic connection to the objective that you're trying to acquire. And during the sleep process, you can actually identify the ideal times and the ideal sleep state during which reactivation is beneficial, and generate reactivation of only the things that you want to reactivate by using those arbitrary links, like playing the sound of a bell or introducing odors that have been paired with your learning objective. And this is really, really cool because this now allows you not only to just generally improve, but also to be very clinical and precise about what you want to improve. And I think it's fun to think about where this research could go if you paired real-time systems with some of the latent diffusion models that are able to decode imagery in the dream state to check whether you're achieving the desired result. If you have a successful reactivation, then you move on. If you don't, then you know that you need to prime that memory again. And so you can actually envision a system that is adaptively checking for things that your brain has learned well during sleep and can move on, and things that you still want to retain but haven't quite locked in yet, and focus on reactivating those during sleep. That type of closed loop system is absolutely possible to do with current technology. No one's done it yet. So off to the races we go. Ken points out that another interesting phenomenon here is that we lose a lot of detail that we learn during the day for various reasons. And you can actually augment your memory capacity by relying on salience, novelty, reward to guarantee reactivation of certain memories. But if there are additional memories that otherwise would be overwhelmed by those other more salient stimuli, you can use TMR to lock those in addition. And this simply shows again that you can do this with TMR. You can do some brain decoding. You observe this entire reactivation process in hippocampus and cortex. It's the same system every time. Okay, we got 5 minutes left. I'm sorry we don't have as much as I would have liked for Q&A, but thank you so much for letting me talk about stuff that I'm obviously really excited about. I get so excited every time I get to go through this stuff. Thanks for lending me your ears. And if there are any questions, I will be happy to take them.
Well, thank you so much, Dr. Coon. That was a very fascinating talk. Thank you for coming and sharing all that knowledge with us. So we do have 2 questions already in the Q&A. The first question is from David. Does listening to an audio recording of the material to be learned while sleeping help at all?
That's the really interesting question. I think the consensus take would be you run into a difficulty if what you're trying to learn is specifically the content of that recitation. We mentioned that this technique relies on arbitrary links. It's important for the stimulus to have no other associations with the thing that you're trying to learn. Otherwise, it interferes with the process. So oddly enough, the results imply that if you want to learn that passage that you're considering replaying during sleep, pairing it with other environmental ambient sounds might be a more effective way to lock it in. Our
next question is from Dana. see, what if you have multiple unrelated tasks that you're learning? For example, learning a new song and a matrix algebra for a class. Are there any studies that compare how sleep improves each of these tasks individually? And does the brain pick a priority, or can it work on multiple tasks simultaneously?
What a fun question. Fortunately, the brain is much better at multitasking than I am, apparently. And yes, if they are independent, non-interfering modalities, they'll get processed independently. You can learn a new scale on an instrument the same day that you learn your organic chemistry memorization. There no doubt are studies out there that test the limits of that supposition, the independence of these tasks. But you can generally rely on each of the stages of sleep doing its job on its type of memory and learning reliably every time.
David just asked if this will be available publicly. The recording, yes, it will. We will post it both to the EP website as well as on YouTube with a link. It will be available to anybody afterwards. Oh, another question, sorry, from Dana again. Does going to sleep under the influence of alcohol have any effect? Does it change anything?
Great question. Any effect? It absolutely does. In fact, the first sleep and circadian study I was involved with was at Brown, and it was studying precisely that phenomenon, the effect of alcohol on sleep. It's really bad for your sleep. It interferes with your sleep architecture. I think there's less that's known about the mechanistic implications of what that does, but it absolutely is known that it really disrupts your sleep, and disrupted sleep is not going to do its reactivation learning job well.
Well, thank you. If anybody has any other questions before we wrap up, please put in the Q&A, or you can raise your hand as well, or type them in the chat, and we'll try to get them answered before we wrap up.
And please feel free to reach out to us over at APL. I do work with students for independent study projects or through classes. We'll be offering the BCI course again in a year from this spring semester, where we touch on some of this stuff. And if there's any questions you have after the talk, please don't be afraid to reach out.
Okay, we have one more question, it looks like. In line with the question about alcohol, do certain spices, eating certain spices, I assume, have any effect when you go to sleep on your retention and your learning?
Yeah, that's a really interesting question. Presumably, yes, if they are carefully paired with some event. I think there's something there. All right. Thanks again, everyone. This was a lot of fun.
Thank you so much.
My pleasure. Take care, all.
Thank you. See you.